Swayam Prabha. Digital India. Educated India.
Welcome back to the second lecture in physiology. So in the first lecture we talked about the basic characteristics what a living system should have, or the foundations what has been laid out during last 200 years, how we define a living system. So there we talked about they should have some basic criteria which includes organization. They should be able to grow and differentiate. They should have some movement feature. They should be responsive to the external stimulus. They should be able to reproduce, and above all, there are irreversible changes which happen in the system. So what today we will do, we will start from those words and define the different terms, anatomy and physiology. So whenever we talk about, if you remember, we talked about this word and I told you to highlight this word, the organization. Okay, so we'll start from here. We'll be starting our lecture 2 essentially from here, from this particular word. Okay, so let's move on to the new slide. So today we are into lecture 2. Okay, lecture 2, I told you, remember that word called organization. Okay. Now, whenever we talk about organization, so think of it. So if you look at an elephant, it kind of gives you something. Okay. If you look about a human being, something like this, or we talk about a small rat or mouse or something, it will be something like this, or we talk about a fish, it will be something like this. Okay. So if you look at these different organizational setups, you see different kind of structures. So all living things have different scheme of organization. So what we use the word is, say for example, these are the different schemes what has been followed by living systems. Scheme of organization, and based on this scheme of organization, they have different anatomical aspects, or in other words, their external structure. So if I had to put it straight across, so it will be like this organization is nothing but is telling their anatomy, and anatomy could be at two levels, their internal structure and their external structure. We'll come to that, internal structure and external structures. Okay. So if we talk about the external structure, you can already see this. This has a different kind of external structure. Different kind of external structure. But inside this, there are different kind of internal joints, internal features, which is making this to walk with 4 legs. In the case of human being, 2 legs, whereas this does not have any leg, it swims, whereas these have 4 legs arranged in a different way. The external features external shape determines its internal structure, and again at what level you are studying it. If you are looking at it, this is called the gross anatomy. Okay, so anatomy itself could be classified into different class. It could be either a gross anatomy, or you could have, if you narrow down your zone of looking, you started looking at the very cellular level, at the cells which are making it, then we call it a microscopic anatomy. Microscopic anatomy. Okay. So the gross anatomy could be at different level. So what are the different level of gross anatomy? Gross anatomy could be classified, or at least could be studied at 4 different levels. First level is the surface anatomy. Surface anatomy, where you studied the surface features. Say for example, in case of human, we are having hairy bodies. In the case of fishes, we have scales. In the case of elephants, there are different kind of a small hair-like structure. Similarly, in the case of rats, you have a fur-like structure. In the case of dogs, depending on which race, which species we talk about, they have different kind of fur structures. So that is the surface anatomy. Followed by, it has regional anatomy. Regional anatomy means different aspects. Say for example, your digestive system of a human being as compared to a horse, or as compared to a cow or a fish. If you do a comparative physiology of these things, these falls under the regional physiology of that particular part, okay, that particular system. Next come systemic physiology. Systemic anatomy, and then comes developmental anatomy. So we talked about surface anatomy, and we talked about the regional anatomy, then we talked about the systemic anatomy, where you see the whole system as a whole, the different systems. Say for example, here you have the cardiovascular system, nervous system, respiratory system, excretory systems. So these different systems, how they are placed along with the other one, and how they interact with each other. What are the different level of their interaction? That's what we studied under the systemic anatomy. Next come is the developmental anatomy, or the developmental changes. So if I take you back to the previous slide, there in the last class we talked about the growth and differentiation. So I told you, over a period of time, as a living system grows from conceiving into the mother's womb, then taking birth, and then going and become aged. So in that process, there are several changes, several anatomical changes which are taking place. Okay? And these anatomical changes started in the mother's womb, starting from development of the body structure, development of the different organs, and then the systems, followed by their crosstalk, and then making a whole system, and then with age how they are changing. So these are all falls under developmental anatomy, the changes during the developmental phase. And this also includes post-developmental changes, which includes the changes which happens post-development, like once you are born. So developmental changes followed by post-developmental anatomy, post-developmental anatomy. So as we grow old, as we are aging, there are changes happening in the anatomical feature at the cellular level, at the bone level. And those are post-developmental anatomical changes which are taking place. Sometimes they are very, very important from the perspective of studying the pathology, studying the development of disease, or how our aging system changes with time, and what are the necessary precautions we needed to take to ensure the safety and integrity and proper functioning of the system. Okay, so that falls under the post-developmental anatomy. Okay, so these are the broad gross anatomy classification. The next comes the microscopic classification anatomy. There we talk about at different level. The first level is the cellular anatomy. When you are studying anatomy from the cellular perspective, that includes different kind of cells within your body. You have nervous cells, they have a different structure. You have a liver cell which have a different structure. You have a heart cell which is a different structure. You have a kidney cell which is a different structure. You have reproductive organ cells which are of different structures. So you can study the anatomical feature from the very smallest functional unit which we call as cell. So cellular anatomy, which is the microscopic level, and the next level is histological, okay, where you take a section of a whole tissue. So cellular or cytology pertaining to cell, histology pertaining to tissue. So what you do, say for example, you want to study the tissue of the heart. So you take a section, small, what we mean by section is, say for example, all of you have seen cheese, or so say for example, you kind of removing a wrapper, or you take a Cadbury or any kind of chocolate. So you remove the wrapper. It's almost like a thin wrapper you are removing from something, and you take that wrapper under the microscope and you study the anatomy. Those anatomical studies falls under histology, histological studies, and they are one of the very powerful tools to give you knowledge about the next level of organization, from cellular to the tissue level of organization, how within the organ these cells are arranged together, what are the channels of the crosstalk between individual cells, and how they interact with the environment. You get an idea about it before you move into the next level, will be this systemic anatomy, which is at the gross level. So if you see, you can study the system depending on, so what is important here to understand, the one critical word, and that critical word is this one, scale. At what scale you are studying the system. If you are studying the system at the cellular level, at the level of the cell, then you are studying the system at micron level. So cytology is, we are talking about micron, because average size of a biological cell, if it is an RBC we talk about, okay, or so anything, it varies from 5 to 25 micron. Okay, a neuronal cell body, say for example a neuron we talk about, which will be around 25 to 30 micron. A standard one, there are ones which have differences, okay, which are much more larger or smaller. So at the level of cellular level, we talk about anything between, I would say, 5 to 20 micron scale. Okay. Next come histology, where you are studying lot of cells in one spectrum. So here individual dimensions will still remain 5 to 20 micron, but you are getting a much more larger area to study. So we move into the millimeter level. Something like, you see the section in front of you, 1000 microns sections in front of you. So you increase the scale slightly higher. Then comes the gross anatomy, where you physically have to take a scale in centimeter or meter, where you are studying these systems. So it is all about scale, okay? And when you talk about development, it is all about time, with respect to time. So when we talk about scale, we talk about space. What level in space? What is the smallest unit in space you are measuring? And when we talk about the changes in that space, we talk about time. So in other words, the real-time anatomy is the study of time and space of a system. At what dimension of the space you are measuring, and at what time you are measuring. Because all this system, if you remember my previous lecture, I mentioned it very clearly. These are all changing in an irreversible manner. In other words, these are all following something called non-equilibrium thermodynamics, where a system is changing permanently. You cannot go back to its original point, because from original point it has deviated. There are changes which are happening into it, and those changes are permanent. You cannot really reverse the clock to go back, that okay, 10 years back this is the situation, I wanted to go back. That will never going to happen. It will be a irreversible system which initially organized itself, and then it started increasing its complexities over a period of time. It developed, it differentiate, it post differentiate, and eventually a point comes and it integrate itself at an optimal energy level, and then a point comes where it disintegrates, what we call as death. And during this whole process, anatomy of the system changes at different level, whether it's at the cellular level, whether it is, even if you can go down, whether it's a molecular level, whether it's at the cellular level, whether it is at the gross level, in other words, which is at the level of the surface, regional or systemic level, there are always changes happening. Okay. So next what we will do, so by the way, this gross anatomy is used by the doctors for another set of classification. We just missed out. Let me put it together again, where they use the medical anatomy is basically the doctors initially look at the gross anatomy. Then there is something called a radiographic anatomy, those who goes through all sorts of radiography tests, and then there is called surgical anatomy. All these three things have clinical relevance in terms of the clinical setting, because a doctor who has to treat you or has to give you, say, some gamma radiation or some other radiation or something to remove a, say, cancerous lump or a mass, they have to know the exact location within the body where they are putting the radiation. Okay? So that falls under radiographic anatomy. Medical anatomy is that how you are defining which part, which tissue, which angle, and the three-dimensional space. So say for example, just you will appreciate it. Say for example, this is how the heart looks like, okay, with 4-chamber hearts. Okay. So now say for example there is a problem somewhere here, or somewhere here. So now this structure is 3 dimensions, something like this. Okay, it's a 3-dimensional structure. Now something is somewhere inside. So with reference to a scale, you have to know in 3 dimension where the problem is. Then only you can target the problem. So those things falls under the medical anatomy, where you should have a 3-dimensional map of a structure, of anatomical structure. You know exactly, you should be able to pinpoint. Say for example, I have to give a radiation somewhere in this. Say for example, this is a mammary tissue or some other tissue where there are cancerous growth. So I should be know, and suppose this is my radiation emitting probe. So I should be able to know exactly where the radiation is reaching. If I move up and down or sidewise, I'm going to damage a whole lot of things, and I may be able to lose track of my exact target, and the patient may die because I'm unable to locate, pinpoint exactly where the tumor is, where the cancerous cells are. So this all falls under the radiographic or surgically. As a matter of fact, even in surgery, suppose I'm doing a laser based surgery, I wanted to wipe out that particular part. So unless I know in a three-dimensional structure where I have to really point the laser, I won't be able to do it. So you realize that understanding anatomy is very, very critical from multiple walk of life. Those who are interested in physiology has to understand anatomy back and forth. Without that, starting from clinical applications to other changes, you won't be able to figure out. So these are some of the clinical classifications of anatomy. Now we talk about a system. As of now, we have talked about a wonderful system, like this whole system. Now this system has certain governing dynamics or certain rules of functioning. So before we get into those rules of functioning, what we will do is we will do a bit of a classification of physiology as a unit. So what are the levels of physiology we can study? Okay. So we talked about anatomy. Now we will talk about physiology and its level. Physiology level. Okay. So you can study physiology at the cellular level. We talked about cellular anatomy. Now we are talking about cellular physiology. You can study physiology at the level of organ, like organ level physiology. Then we can study at the systemic level physiology, or we can study at the pathological level. So these are different levels where you can study it at cellular level, systemic level, or at pathological frontier. And when you talk about the organ level, this is also sometimes they use a word called special physiology, where you talk about, say for example, I want to study the cardiac physiology. Okay. So I'm exclusively studying this. So when we talk about the cellular, say for example, within cardiac physiology, I wanted to specifically study the cellular nature of say cardiac myocytes or ventricular myocytes. Okay, we will come to that. Do not worry about these words. Or within the kidney, I wanted to study the cellular structure of a nephron. So that falls under cellular physiology, or a systemic physiology, where you are seeing the interaction of different systems, or pathological physiology. This is one way you can classify it. So then what you can do, what are the level of organizations? If you remember, we talked about the organization level. So let's draw the organizational chart. Level of organization. Okay. So the level of organization could be organization of system. Could be at multiple levels. The first level what we talked about is chemical or molecular level, which is the top of the pyramid. Chemical or molecular level of organization. Next is cellular level of organization, at the level of the cell. You can study a cellular physiology. Third higher organization is the tissue level of organization, where you talk about the histology. Fourth is the organ level of organization, where the different tissues form an organ. Next is when different organs interact with each other, organ system physiology. Okay. And final one is the organism level of physiology. And if you draw the pyramid, it will be something like this. Okay. This is how the pyramid will look like. Okay. So now, at what level you are studying it determines what kind of information you are going to get. You can study at these different levels, you can study the anatomy and physiology. You can go even further deeper, interaction, where the current trend is to study at this level, somewhere at the crossroad, the chemical and the molecular and the cellular level. Okay. Next level comes if you are studying the physiology at the tissue level, anatomy and physiology. Third, at the organismic or at the organ level. Fourth one is the interactive level, organ system level, and the final one is the organism level. The whole system you are studying in one go. So whatever level you study, you have to realize that you have to correlate it back in that pyramid from the chemical to the molecular aspect all the way to organism. So this is how the system works. Now there are certain governing rules. So there are different level of control systems which are happening at different level, at all these different levels, and those are called different feedback mechanisms. So what we will do now in the next class, we'll discuss two aspects. One, how a system responds. What are the basic thumb rules for a system to respond? And once it responds, how it react to it. In other words, it has a responsive element and then it has a reactive element, and we'll come to that. What are the different feedback loop systems which are functioning, which includes a negative feedback and a positive feedback. After that, what we will do, we'll study about the reference. Say for example, what I meant by reference is, say for example, I have to study a human body. So what will be my scale reference? How I'm going to study it? Will I study, say for example, you look me from the front. Okay, there are certain features you see in my body. You look me from the side, you get certain feature. You see me from the back, you get certain feature. So all the organs, say for example, this is one organ. Now depending on which angle you are looking at it, you are looking from that side. My viewers are looking for that side, I am looking from the back. Say for example, I keep it like this. Okay, for you, you see it's a, there's nothing. But if I show you the back, it is like this. There's a huge hole out here. So from which angle you are studying it? Are you studying it like this? Are you studying it like this? Are you studying it like this? Are you studying it from my angle like this? So these brings us to different planes, sagittal plane, coronal plane, and we'll talk about that, how you are studying the system. In other words, that brings us to the basic fundamental of what is the scale you are following, how you are determining the scale, and where you are positioning your reference point. What is your reference point of looking at the system? Are you looking at this angle? Are you looking at this angle? Are you, dissecting the system like this, or you are dissecting the system like this? So we'll talk about next two things. We'll talk about the feedback controls of our system, and then we will talk about the different sectional geometry by which you can study a system. And that will pretty much brings us the point that we can initiate to study any of the organ systems. Okay, thank you.